Good morning, Dr. Ujjwal. Hello, Ujjwal. Can you hear me?
Yeah, he has to unmute himself.
Ujjwal, can you unmute yourself?
Yeah, we can't. Yeah, we
Hello,
can't hear. Yeah, we can hear you now.
Good
Hello,
morning.
Dr. Ujjwal.
Yeah.
Raman here.
can you hear me?
Yes, we can hear you now. We can hear you. I think he can't hear us.
Yeah, now I can hear you.
Okay, great. Wonderful. So, welcome to ACBR seminar series. So, I think without any further delay, we can start. Sanjay?
Sure, ma'am. Sure, ma'am. Please.
Because all our speakers and guests are here. So we can begin. So will you start or Dr. Sanjay?
Yes, ma'am. I think you can start. So, good afternoon, everyone in India, and good morning to our speaker for today's lecture, Dr. Ujjwal, who is in Sweden. I'm pretty sure many other faculties from his institute is also attending today's talk. So with this brief introduction, may I now request our director, ma'am, Professor Daman Saluja, to kindly introduce ACBR and also introduce our chief guest. Thank you, ma'am. Over to you.
Yeah. Hello, everybody, and it is really nice for Dr. Ujjwal to give a webinar in this webinar series of university. Though COVID has really restricted our movements, but it has allowed us to have speakers from all over the world. And we are really thankful to Dr. Ujjwal as well as to our chief guest, Professor Bamzai, and chairperson, Professor Natarajan, to agree for their key roles in this seminar series, in today's seminar especially. Most of the people who are attending knows about ACBR, a part of the Delhi University, but some of the new students and maybe Dr. Ujjwal, I would just say a few lines. We are located in the North Campus, um, and the mandate of this center, which is still not in the form of a full department, but center, as it is quite small, is to really work in an interdisciplinary manner and to come out with uh, you know not only basic research but also translational research, so that we can contribute something to the uh, health sciences or medical sciences, as biomedical sciences as the name has been put. So everybody in the center is working towards various areas like cancer, neurobiology, drug discovery, basic molecular biology, molecular immunology, genetics. So different labs are doing this in both in independently as well as in the integrated manner. So we have lot of intra-departmental collaborations in addition to collaborating with other institutes. to work towards getting something meaningful out of the research that is being carried out in this institute. In addition, we run MSc program here and PhD program, and the BSc program is run in the colleges. There are 3 colleges where BSc Biomedical Sciences is being taught. With this brief introduction, I would like to introduce our chief guest of today, Professor R.N.K. Bamzai. He really needs no introduction, and because of his huge amount of contribution to the health sciences, biological sciences, life sciences, but to all the new students of MSc who have joined recently and PhD students of ACBR, I would like to introduce Professor Bamzai. First of all, I must thank him for agreeing to to be the chief guest of today's seminar, and also to spare time, because he is very busy, and he says, I will go through his contribution, you will realize that he is a very, very busy person. Professor Bamzai did his PhD from AIMS, and then he started as a faculty in IMS, Institute of Medical Sciences at BHU, and then later at Jawaharlal Nehru University, Delhi. And he has expertise both in life sciences and he was also Dean of Life Sciences and Computational and Integrative Sciences at JNU. He was coordinator of National Center for Applied Human Genetics at JNU, at SLS and Bioinformatics JNU. Professor Bamzai has received the most prestigious award, Padma Shri, which is given to him for his contribution to science and engineering. In the year 2012, he was given Padma Shri. So that summarizes most of his, achievements and the awards. But to add, he is member of all the he's fellow of all the academic societies of India, National Academy of Science, and NASI, NAMS, INSA, and many of the international academies. He has been editor of some of the journals. He has been member of various task force committees and on the review boards and task force, and chairman of several task force committees, and I can go on and on. He has guided 40 PhD students, published almost 150 papers to his credit in very prestigious journals. And currently he is even after, he was also vice chancellor of Shri Mata Vaishno Devi University at Jammu. So I can go on, and I would just say that even after his retirement, he is contributing a lot to science and technology in India by serving at many of the committees of DBT and DST. And currently he is also honorary director of Delhi School of Public Health at Institute of Eminence, University of Delhi. And his concern in public health and creating a genomic literates within the society has really resulted in establishing a genomics and public health foundation in Dehradun, which he has established. And he is contributing a lot to the starting up of the Delhi School of Public Health in Delhi University. He is the chief driving force behind it. With these few words, I would request Professor Bamzai to say a few words on the area of system biology and the gap areas and words of wisdom to our students, to our faculty. Professor Bamzai, please.
Sir, can you please unmute? Sorry.
Oh, I'm sorry. Good afternoon, all, and good morning to Ujjwal. So let me begin with congratulating Dr. B.R. Ambedkar Center for Biomedical Research, its director, Professor Daman Saluja, who happens to be the organizing secretary for this event, and the convener, Dr. Sanjay Kumar Dey, of the Centenary Seminar Series, University of Delhi, for organizing ACBR virtual keynote lecture by Dr. Ujjwal Neogi of KAROLINSKA Institute, Stockholm, Sweden, under the chairmanship of Professor K. Natarajan of ACBR. Well, over the past decade or two, you all know, researchers are generating and attempting to integrate large amount of multidimensional data generated through a wide spectrum of high throughput omics techniques, and to make a comprehensive assessment and derive useful application for understanding molecular mechanisms and functions, customization for individuals and for society as personal and precision medicine, new informatic approaches, systems theories, including network analysis, machine learning, and AI, are applied to understand relevant and complex biological phenomenologies and computational theories. Well, we have had insight. We are going to, in fact, have insight from Ujjwal on the progress, which the title he has given of multiomics systems biology analysis to understand progressive immune disruption in COVID-19, very relevant in current times. Well, welcome, Dr. Ujjwal, and we are looking forward to hearing you and not reiterating what you will be speaking and touching upon, but I feel the point of heterogeneity in clinical presentation of a disease and the severity so unique to each patient or afflicted individuals, converging on key tuners within a cell could then be targeted for intervention in any disease state, which is a challenge to reckon with. Well, the need to put all the layers of different omics information to generate a network matrix to choose from remains a good bet. And I'm sure Ujjwal is going to treat that information well with the immune readouts, which he has worked on in detail with multi-omic approaches, and with the informatics knowledge and tools at the moment which are available with us. Well, all of you.
Thank you, Professor Bamzai, for your words and wise words, and giving direction to the youngsters, also the coming up areas of system biology, omics, multi-omics approaches in solving disease-related problems. I would now like to request Sanjay to take over.
Yeah, thank you, ma'am. So may I now request Dr. Meenakshi to kindly introduce our chairperson?
Yeah. Thank you, Dr. Sanjay. It's my immense pleasure to introduce our chairperson for today's seminar, Professor Natarajan, because he is the first person I met in ACBR when I entered in March 2010. We both were sitting at reception waiting for Daman ma'am. He was joining as professor and I was joining as postdoctoral fellow. We introduced ourselves, and today I have the privilege to introduce him to the community. So he is serving in ACBR from 2010, as I told you. He joined as professor. And before joining ACBR, he was the principal investigator at ICGEB, and he was leading the immunology group there. And he has served as director in ACBR also from 2013 to 17. He completed his PhD from University of Delhi in 1994. And then he did his postdoctoral research from MD Anderson Cancer Center, Austin, Texas, USA. And his key research areas include immunology of infectious diseases, including mycobacterium tuberculosis, HIV, Plasmodium, Streptococcus pneumonia. He is an elected member of several reputed societies, including Indian Academy of Sciences, Molecular Immunology Forum, Indian Immunology Society. He is also the member of several task forces for research funding, members of selection committees, and he is an editor of several internationally reputed journals. He has already published more than 100 research articles in reputed journals, and he has supervised several PhD students. And I don't have words to describe about him. And personally, he's a jolly person. I will never fail to mention this every time. And with these few words, I would like to thank our today's chairperson, Professor Natarajan, for accepting our invitation and making our seminar series successful series. Thank you, sir. Now the stage is yours, all yours.
Thank you, Professor Daman, Dr. Sanjay, Professor Bamizai. Always a pleasure to see you virtually and in person last month. And I welcome Ujjwal Neogi from Stockholm virtually. Hopefully in the next year seminar series, technology will take us in not having a 2D but a hologram, just like how we have been like our Prime Minister unveiled Netaji's statue. So maybe the technology will drive us, and maybe systems biology will also contribute in its own sweet way in this area. And yes, of course, Meenakshi, I always remember my first day of meeting you, and then we were both waiting for Dr. Daman to join for our various joining formalities. And okay, that apart, it is my pleasure to welcome a young faculty, Dr. Ujjwal Neogi from KAROLINSKA Institute, Stockholm, Sweden, who would enlighten us in the next 1 hour or so, I believe, on systems approaches to studying immunity in COVID. And I think I must congratulate Dr. Daman, Dr. Bamizai, and of course Sanjay for choosing Ujjwal as one of the key speakers. This talk could not have come at a better time. This is a time when, like COVID, the SARS-CoV is mutating like crazy. It's throwing up mutations left, right, and center. So the point of concern, or the point of investigation, I would rather say, is to see how our immune system going to interact with it, how are vaccines going to act on it, how much protection, and what kind of protection. All these things cannot be answered in a reductionist approach. One has to look at the systems level. And who better than Sanjay as a young dynamic faculty? I was just going through his CV, a brief CV, and was very impressed by the way he has actually matured into a fine researcher at this young age. So let me begin the formal introduction. So Dr. Ujjwal Neogi is an associate professor at Karolinska Institute, Stockholm. He did his PhD from the same institute in 2030. He's a fairly young fellow in translational genomics of HIV subtype C and in India. And he did his 2 postdocs, one at St. John's Research Institute in Manipal as a Wellcome Trust India Alliance Fellow, and then later on, I believe, in Karolinska. And he's been there ever since, and he specializes in looking at a problem from a holistic point of view using systems approaches, something, I'm sure the young audience for today, the young MSc and PhD students would appreciate and understand that one has to look at a multiomics approach and from various angles, just like Professor Bamizai outlined very nicely in his little introduction. And he did his postdoctoral work on SARS-CoV-2 and HIV. HIV, not SARS-CoV-2. He's an editor of several international journals. He has a number of awards to his credit. And he is also a adjunct professor at University of Missouri in the United States, and also a guest professor at the Manipal Institute of Virology. So he has a long way to go and many more accolades to achieve. And we all wish him all the best, and our good wishes are with him. So without any more ado, let me welcome Dr. Ujjwal Neogi once again. I request him to start his talk. Ujjwal, all yours.
Yeah, I'm trying to share because I am not much used to with this Google Meet. So let me see if I can.
So Ujjwal, you can see one arrow. in the bottom.
Yeah, can you see?
Yes, now it's coming.
It's come. It's come.
It's in the power full mode, right? Or
Yeah, it is in the full mode. It is in the presentation mode.
okay. So thank you, but I cannot see myself. Okay, thank you for this nice introduction. And
You can't see your PPT?
no, maybe I just stop presenting and restart.
Yeah, restart. You open your PPT first and then share it.
You can see now, right?
Yes, we can see now. Can you
Okay.
see it?
Yeah, now I can see.
Okay, great.
Yeah, so thank you very much for inviting me, and I would like to thank first Sanjay, whom we just started collaboration, and hopefully we'll have go, and Professor Daman and Professor Bamizai and Dr. Natarajan to introduce me in a nicer way. So what I'm going to talk today is mainly our work on SARS-CoV-2 that we started in April 2020. Because before that, I was mainly working with HIV and HIV functional cure. So what I'm going to talk today, how we use advanced multiomics system biology studies and to understand the system level, because that's much easy to go for to understand how our body respond when we have any viral infection. So basically what my research group here in KAROLINSKA, what we do, we just try to identify host metabolic reprogramming following infection. Why this is because virus is obligatory parasite. And virus need our body to respond to the need our body to get energy and replicate. So that's why it's much be easy to stop the virus from if we stop the energy from them. So first thing what I started with HIV, that is because there's several genetic diversity of HIV. The antiretrovirals are working, but there are lots of drug resistance mutation can come. And this is actually a common feature for most of the RNA virus, including SARS-CoV-2. Now every second day, we are hearing that there's a new variant. But the approach, what we are targeting, it's mainly the metabolic pathways that virus uses, so that we are less concerned about the mutations. And then my colleague Shoham Gupta is trying to link this metabolism part with innate immune response, because he is also an Indian, and he joined my group as assistant professor. The virus we are looking is dengue, chikungunya, CCHF, like that is Crimea Congo hemorrhagic fever virus, HIV, and more recently with SARS-CoV-2. And the pathway that we are mainly working with this central carbon metabolism. So I will go through in details in the later stage, but just to give you a brief, that we need a virus when infect, it's change the properties of several RNA virus, like PIK3, AKT-mTOR pathway and HIF1 signaling, as well as glycolysis, gluconeogenesis, and fructose mannose metabolism. How do we do our research? So what basically we do, we start from the cohort, because that is the base of our study. So we try to define with our great clinical collaborator, okay, what is they are seeing in the clinic. So we have cohort, and then we generally get different kind of material like whole blood, PBMCs, plasma, fecal material. And we go for different levels of omics like transcriptomics, proteomics, metabolomics, lipidomics, microbiome. Similarly, when we know what kind of modulation is happening, then we go to in vitro model. So just to model the disease, how it's taking the energy, how it's going. So it's mainly a longitudinal studies, because it is difficult to get pure acute stage infection patient, because as the patient will give or show any symptoms after a few days of therapy, a few days of infection. And then if we succeed, recently I'm moving to the animal model because I am not much fan of animal experiment, but now we have started with SARS-CoV-2 with ferret because I'm part of a vaccine study. For CCHF, we are initiating the mouse model. And for HIV and SARS-CoV-2, both monkey. Here also it's a mostly vaccine oriented. Then when we have this different layer of omics, we try to integrate because I am not again much fan of p-values between one omics, rather how different network or the network of genes or metabolite they are working. So we try to model context or disease specific models. So what are those? Those are mainly that when we say HIV patient, maybe with 10,000 viral load in the body, and then they are not responding to therapy, and they are maybe elite controller. So this specific disease as well as specific individualized model. So the method we use, which is presently available, are mainly the similarity network fusion, weighted co-expression network, and multiomics factor analysis. Then if we get a target or a hit, we go for mechanistic studies. So at present, as I mentioned, major focus is on human immunodeficiency virus or HIV, where the cohorts are from Sweden, Cameroon, India, and Denmark. Then I started 3 years before CCHF, that is Crimea Congo hemorrhagic fever, and the cohort is from Turkey. Then we started with coronaviruses when SARS-CoV-2 came, because my lab was the third lab in Sweden who started growing the virus or isolating the virus and beginning of the pandemic. So we have cohort from Sweden, Italy, and Ireland. And recently, actually, it's tomorrow, there is a task force meeting where I took part as a co-investigator from Manipal Institute of Virology on dengue. So that's from India. So the major theme of my lab is starve the virus to death. That means don't allow the virus eat the energy. Rather, it can be stopped to so that they can die off. So for today's talk, I'm not going so much background in the SARS-CoV-2 because we know a lot. There are lots of presentations, there are lots of media event that happen to know how the virus is going, what the different variant of concern is coming. Rather, I will go for our study. That is, the first one is the metabolic perturbation associated COVID-19 disease severity and SARS-CoV-2 replication, which accepted in 2021 in molecular cellular proteomics. Then, as Professor Bamze said, that when a virus hit, the response are different from different individuals. So what we did in the second paper, which is actually currently under review, but it's available in the bioarchive. So that is personalized model to understand the immune disruption. So here we develop individual model for individual patient and then try to see what is common in one patient, one group of patient. And then the last one, this is again unpublished data from our Ireland cohort, where I was mainly involved in the metabolomics part. And then to see how gut microbiota can play role in excessive inflammation and COVID-19 case fatality. So this paper is also online in bioarchive. So the first question that when the virus come and when the COVID-19 came, what we try to identify, what is there any similarity to any disease? To our surprise, when we started looking into it, it's a very nice article came based on the initial data from Italy, Spain, and China, the similarity between COVID-19 and metabolic syndrome. So few examples is like the subsequent post-acute sequel of this COVID-19, there's a heart attack, which is common in metabolic syndrome and type 2 diabetes, which is also have been observed in COVID-19. There is also elevated liver enzyme, which has been both seen in both the cases. There are lots of neurological complications like damage to blood vessels, nerve caused vascular cognitive impairment that has been observed in metabolic syndrome. Also, we have observed seizure, stroke, and then there are several neurological complications that we have observed in SARS-CoV-2. So then this also came the same paper, perspective article. They have also shown that what is the long-lasting effect of COVID-19 on metabolic health. So muscle atrophy, then altered glucose metabolism, so which is one of the important factors, and that also we are looking into our thing. Change in fat compositions, increased risk of future cardiovascular event, and like similarity like depression, anxiety, those who are observed in the long COVID patient. So when we started our study in early 2020, we started to look what kind of metabolic reprogramming happen when virus go inside the cells. So during that time, two nice study came up. One, it shows that hypoxia and HIF1 stabilization can lead to disruption in the heart, lung, and blood vessels, and can be responsible for respiratory and circulatory disturbance and failure. And also the second study that came up in cell metabolism, where they showed that when SARS-CoV-2 infect, they increase the HIF1. So that could be a reason for the cytokine storm, because there are several cytokine or ISGs, interferon stimulating genes, are controlled by the HIF1 when it translocate to the nucleus. But one thing was a bit puzzling, that which cells it can infect, because when we started we started with lung cells, but we are not able to see any infection. So during the first phase, we have published that data already, that what are the different cells that SARS-CoV-2 replicate. But one thing what we identified, that HU7, which is can be infected by the SARS-CoV-2. So we did, because to model a disease, we need a longitudinal data. So we did a longitudinal infection and look for 72 hours up to. And then we did transcriptomics, we did proteomics, and then we do proteotranscriptomics co-regulation. So one can see the viral RNA in the supernatant is going high over the time, and also inside the cell, what is the composition of different proteins. So it's mostly stable. And we came to know in the later stage that the replication is very, very slow in HU7, and we did not see any cytopathic effect. Similarly, there is also increase in different viral protein. So that can be identified using the proteomics study. Then we use the weighted co-expression network. What is that? That is generally to identify how the network of genes are working together. So here we are not interested in single gene or single molecule this way, but rather this is a cluster of gene. And when we do, we give a weight to each of the interaction, and based on the weight, we can define different clusters of things. So for example, in the transcriptomics data, we identify 5 networks. And then based on which network is control the other networks, we define that as a central network. So for example, in transcriptomics, that's community 3 or is the central community, which also controls the other 4 communities. And when we did gene set enrichment analysis, we identify the central community, the majority of the genes are from mTOR signaling pathway, MAPK signaling pathway. The similar thing we did it with the proteomics, and here we identify community D is the more central, and this proteomics is actually mainly the antiviral response, like ubiquitin mediated proteolysis, N-glycan biosynthesis. So at proteomics and transcriptomics, the regulation is different. Then we try to simplify the analysis. So as you see, there are 5 networks, the transcriptomics networks are quite interlinked with each other. And these are the pathway that we are interested, that HIF1 complement coagulation, because we have seen blood coagulation in this patient. Then we have TNF signaling, ERB signaling, etc. So what we have seen, the communities are quite interlinked at the transcript level, but not that way, the protein level. But community B, C, and D, where the majority of the genes from different pathways are there, are quite interlinked. So this way one can see how the different pathway are interlinked with each other. Then my group was working in the HIF1 signaling pathway in HIV. So we have the antibody that time, and we have observed, particularly the phosphorus 6K1, you can see nicely increase over time. HIF1 is a different modulation. First is decrease over the 24 hours and then increase at the 48 hours, because there is a negative feedback loop happening. And when we see at 24 hours, you can see there is the virus has used of HIF1 thing. So that indicates that there's a most of the, so this is RNA scope. So we detect HIF1 RNA. So when this HIF1 RNA is less, that means there's lots of translocation in the nucleus so that it can produce the protein, and then the glycolytic modulation happens. So I will show you in the next slide. So this is the paper we identified, but also what we did in this study, we tried to block this pathway using pharmacological blocking. So can we identify any specific pharmacological compound that can stop the viral application? So we identified there's a moderate decrease in MK2206 when we block the AKT, which is one of the, this is the complete pathway of mTOR HIF1. But the striking feature is when we block the S6K1, there is S6K1, this pathway, and also the activation through the SAHA, there is no change despite the virus is using. So we don't know, but when we use the activator, and we can see there's a huge increase in the viral load. So though there's several study in the beginning say use mTOR inhibitor, we are not much sure, so we are not given a complete picture, because if there's something goes wrong on off target, the replication can go rapidly. In the next paper, we went to see the metabolic reprogramming in patient, because as I said that it's very important to know what is happening in the patient to guide our bioinformatics data, because bioinformatics data is all predictive. It's not absolute. And what happens in the cell line, basically in the cancer cell line, may not reflect in the patient. So there is a hospital in Stockholm, it's called South Hospital, which is an infectious disease hospital. From there, we choose patient, those who are mainly hospitalized, because I think you might know that Sweden initially do not even tested, and there is no lockdown for us anytime during the whole pandemic. So we never had any lockdown, and only they were testing the patient in the beginning when they reached to the hospital. So we first choose the hospitalized patient based on oxygen consumption, because that time WHO has not come up any disease severity grade, and we choose patient 29 who are, we called it hospitalized mild, those who have O2 level less than 4 liter per minute. And then we got severe patient, only 12, who has more than 4 liter per minute consumption. This is also a reason for the small sample size, because as we do lots of metabolomic study, we are not willing to take patient when you started the glucose or any other amino acid as a supplementary treatment. So we try to collect the sample the day of hospitalization, or at least before they go to the ICU. So this way we are not able to choose nearly 3 months to get only this many patients. We also identified negative controls, those who came to hospital, either the family member of this patient or study nurse there, those who are PCR negative for COVID-19. So like that, we identified 31, but when we tested for antibody, we found 10 has antibody. So this way, we call this 10 antibody as a convalescent and 21 as a pure COVID-19 negative. And this was very early. So the sample was collected in April 2020 to June 2020. And as expected, it's difficult to match the healthy control because initially most of the patient are very old age, but we try to match between hospitalized mild and hospitalized severe. And you can see, so the last column is between hospitalized mild and severe, not much difference apart from the oxygen consumption, because that was our inclusion criteria. So first thing we did a plasma proteomics profile, this method called proximity extension assay. And it's actually done by my collaborator, Professor Ulf Landgren, who is the inventor of this methodology. So what we can do in this method, we can just use 1 microliter of blood or plasma, and you can quantify 92 proteins simultaneously. So you can see it here. So these are the proteins that are at least differentially regulated, at least one compression, and you can see that healthy controls and mild and convalescent are more green. That means their level for these particular proteins are low, but lots of red in COVID-19 patient. And so some of the key was IL-6 that we know, the cytokine storm, what we observe, and also IL-12, which is the only interleukin that has came down. Next, we look for the metabolic profile. So this metabolomics study is done on plasma, and the data came from actually 4 different platforms. And the method is ISO 9001 certified. So you can clearly see the healthy controls or healthy COVID-19 negative patient and the positive patient are clearly separated. And most of the difference are actually in the lipid and amino acid. And when you look for the pathway to what we have actually seen in our cell culture data, we have seen a similar kind of pathway, but not exactly the same pathways. And then 2 things what we is striking to us, one is the glutaminolysis. So glutaminolysis is nothing but the lysis of the glutamine, and what we have seen, there is a gradual increase in the glutamate level and then decrease in the glutamine level in the blood. In the same way, the glucose and mannose is also changed, and you can see the mannose actually is a good biomarker even to distinguish between severity patient. And that is what we have proposed, that mannose is a good biomarker. But if mannose is high, one of the factor could be the mannose binding lectin should also be high. So during that time period, while we are doing our study, a study came up and they showed that COVID-19 patient actually high mannose binding lectin. And we observed the same trend in our cohort also. But one surprising thing is those who are convalescent, they also have high mannose binding lectin. So this also can serve as a good marker even for the convalescent patient. So in take-home message, what we have observed is glycolysis and gluconeogenesis is used by the virus. Then fructose and mannose metabolism is also used by the virus. Glutaminolysis is the 3rd pathway that use, and glucose mannose are also the determinant of disease severity.
Ujjwal,
So,
do you mind taking a question?
Yeah, sure.
Okay, so I have one comment and one question. You talked about MBL increasing in convalescent as well as in severe cases, right?
Yes.
So the distinction is based on the quantity, or is it at the time? Because convalescent, you said, are the ones who have possibly mild infection and recovered. And jointer to that is like also looking at the alternate pathway of complement, because MBL does play a role in that.
Yeah. So yeah, time also matters because we don't know for this convalescent patient when they got infected, because in the beginning of the pandemic, we in Sweden, we are not testing anyone who has actually mild symptoms. So we don't know when they get it, but the basic thumb of the rules is particularly there are a few doctors and nurses are there. So at least there should be a month apart from any like even mild symptoms. And for the severe cases, this is the first or second day of hospitalization. And generally, like it takes in the beginning, 7 to 15 days to see any symptoms and reach to the hospital. So it could possible the time is also important. But what we have seen here, rather than mannose binding lectin, which was proposed by this Erickson et al. Study, we see the mannose is good biomarker for disease severity. But mannose binding lectin can go up and it can stay little bit longer, even in the convalescent patient who has mild or no symptoms at all. Is that answer your question?
In a manner, because if mannose is increasing in the plasma in severe cases, something must be done to get it. No, like the system must be responding it by increasing mannose, and then the best is to look at bind it to mannose binding lectin, and then go for the complement pathway to get rid of the RNA that may be circulating. So in the severe cases, it makes perfect logic. I'm surprised at the convalescent. That was my point.
Yeah. And if you see even the convalescent, though is P is less than 0.02, it's like more or less that most of them are the upper quartile of the healthy.
That is why I asked is it the concentration or the time are you looking at?
Yeah, it
So
could be the
both.
concentration too.
That's okay.
Yeah, because these is microgram and these are the ELISA. Because mannose binding lectin, we did it on ELISA, so at least we know the concentration. But other are actually arbitrary value, so it's relative to others. Okay, so to continue with that, so what we are actually looking is mainly the center carbon metabolism, because these pathways are interlinked to this. During the same time, there is a nice paper came by Cordova et al. Cell metabolism, which is one way for us as a virologist is controversial, but they have actually initially shown why there is a more viral replication in diabetic patient, because they have proposed high level of glucose actually is a cause of high viral replication. But in this study, what they infected is monocyte, which is quite striking for us because we were struggling to infect any monocyte and see productive viral replication. Because what they did is they give glucose, pyruvate, galactose, and 2-DG. And 2-DG is a drug, I think it's already been used in India in severe cases. And they have shown that when there's a glucose there, there's productive viral replication. So we also try to look what mannose play a role, but to our surprise, in the HU7, in the Kaluthi, that is a lung environment, what we have observed actually high glucose block the viral replication, and there is no change. Maybe it's a residual virus, no change in the viral load. Then subsequently, there's a study came up that they have shown that SARS-CoV-2 do not it can infect the monocyte, but this it cannot produce the replication competent virus. That is, the infection is abortive, and same thing for macrophages also. So I think it's need to be this data need to be carefully looked, but as they have clearly pointed out, I think they are the first to show that 2-DG is also can block the viral replication. The next thing what we want to look when we have the glucose, mannose, and glutamate as a 3 good marker, does the transporter in the blood cell is changing? Because glucose and mannose is used by GLUT1 apart from other GLUT transporters, but GLUT1 is the one of the good thing that there's a good antibodies available, and XCT, which is for the glutamate antiporter. So that means it throw out the glutamate outside and take cysteine inside. So what we have observed in CD8 T cell, GLUT1 is high, but XCT, the glutamate transporter, is mainly high in monocyte, classical monocyte, intermediate monocyte, and non-classical monocyte, and to some extent more higher in both the COVID group, both hospitalized mild and severe. Then in the first study we use HU7, but now that by the time we are able to identify how to infect the lung cell model, so one is, so we use CALU3, which is a lung epithelial cell model, and infect the virus to that. And after 24 hours infection, we did a proteomics, untargeted proteomics study, and what we have observed is like there are increase in pentose phosphate pathway, which is again part of the center carbon metabolism, but decrease in oxidative phosphorylation citric acid cycle. And you can clearly see a very distinct pattern of those pathway. So this, and if we make it a pictorial thing, we have observed that most of the cytoplasmic part, the proteins are upregulated, but the mitochondrial part is downregulated. And also the part of where there is the TCA cycle component in the cytoplasm is also increased. So during that time period, it has already shown, or there's some perspective article that there is a mitochondrial dysfunction. So we just look the total mitochondrial DNA copy number in blood cells, and you can see there's a little decrease in the mild patient. It's just p values which matters, but there's a little decrease in the hospitalized severe cases. So then in the same cell model, we block with 2DG. And when we are doing this experiment, I was very happy that DRDO and Dr. Reddy's has came up with this therapy for severe patient. But our major interest was glutaminolysis. So we use 2DG as a positive control that it will work, because in our hand, none of the drug that time was proposed, like remdesivir, hydroxychloroquine, was working to inhibit the viral replication. So, and then we use DON. So DON is actually very not specific to one enzyme, but it can work in multiple enzymes of the pathway of glutaminolysis. And what we have observed, there is a drastic reduce in the relative RNA copy inside the cell. So that give a hint there is not
Ujjwal,
much.
Sorry to disturb you. So you optimized any dose, or you arbitrarily used any specific
No,
dose?
we optimize the dose.
Okay.
So the dose which is not toxic to the cell, we used. The highest dose that were not toxic to the
And
cell.
by that time, it was already approved in India, or no?
Yeah, it was, I think, approved in, like when our paper came out.
Okay.
By that time it was approved, but I exactly don't know when it was approved.
And who discovered this DON? DON was already known, right?
Yeah, DON, I think it's Mark initially.
Okay.
But this, like now there is a study going on in HIV. There's a form of DON, so that is called JU839,
And
which
here
is,
you
I think,
see
the third.
higher effect for DON. That means if you reduce the dose, could have the similar effect. That also you checked, or no?
Yeah, we checked
Okay.
these things. So
Okay.
this is the dose we use where at least 90% cell are alive, because
Okay.
these drugs are quite toxic.
Okay. Thank
And
you.
so, okay. And then, but there is a wider load, so it could be residual because it's a similar, though there's a little bit change. It's more or less similar to the 2DG. And now what we are doing is actually we actually went for the animal model. So hopefully in a couple of weeks time, I know whether this DON saved the mice, this K18 knockout mice or not. So this next study, what we did in the same cohort, we did RNA-seq because it took some time to get.
Ujjwal, can you go back to your previous slide?
Yeah.
So this, I'm talking about this viral load versus relative RNA copies. So the RNA copies is down, but your viral load doesn't seem to be going down as much as your RNA. Because at what time point was this experiment terminated?
It's 24 hours post-infection.
So if you let it go for, let's say, 48, 72 hours, did you see a relapse? Because that could also
No,
explain
not
some amount of
much.
long COVID. Huh?
Not much actually. Because what happens these 48 hours, we see quite a lot of cell death.
Oh,
So
because
that's
of
why
this toxicity. I see.
yeah.
Okay. Okay. Well, I was wondering whether it'll be a relapse because it's down but not out kind of a thing. So
Yeah.
it may
So
come
this
back
is inside
as
the cell. So
yeah.
replication is stopped. Maybe these are the residual which came in the supernatant.
Ah, I see. Okay. Okay. No.
So next we did transcriptomics because during that time it was quite strained in the facility where the central facility where we do transcriptomics. And then we use the same proteomics data, same metabolomics data, and also we did flow cytometry, and we did integrative omics thing. So first thing we did is the digital scene quantification. This we have developed now. So we use a algorithm, deconvolution algorithm called EPIC. The EPIC was used actually as a background of cancer cell line. But we have actually blood atlas published last year from Karolinska Institute. So we use the blood cell markers and its expression value and using the EPIC tool to do a digital cell quantification. So with this quantification, we identified that neutrophil is high, which is known. Classical monocyte is high. Gamma delta T cell is actually impaired in hospitalized severe patient as well as mate cell. So then we are interested to look. We are interested to looking into how the coordination between these these cells working. So you can clearly see. So its coordination means its association, and again we give weight, not as a simple association. And you can see in the severe patient the crosstalk between cell are going down. But again, these are prediction. These are not true. So then we develop a panel because our major interest and changes we observe in the monocyte panel for mononuclear phagocyte, and which can identify different subset. And we see the similar trend, like classical monocyte is high, intermediate there's no change, but there are several patient has low intermediate monocyte, and also the DC population which are go down. And when we check the crosstalk between or the association between this cell population in CD4 and CD8, so the changes are mainly happening in the mild and severe, not in the healthy. We
Did you
also
look at
look.
plasmacytoid dendritic cells, PDCs, in the lung? Because they seem to go up in most infections to
This is in blood.
immune suppression.
This is in blood. We don't have lung.
Yes. Okay.
Yeah.
Okay, fine.
Yeah. Okay. And then we also look for CCR5, CCR2, and the co-receptor because these are important in the trafficking. And we have seen the CCR5 is high in classical monocyte, but severely low in a severe patient. So it can be a dynamics, or the time has very important role to play. And why this is low could be like it's only the immune system is impaired in those patients. The next thing what we did, because all the COVID patients, they are not, despite they have severe, they have the comorbidities, they are not responding that bad. So what we develop a similarity network fusion. So what this is basically you take one omics or second omics or third omics. When you do analysis only based on one omics, you can see the patient. So these are the patient versus patient clustering. They are not that strongly clustering. Similar here, but when you fuse these 3 into 1, you can nicely see there is a nice cluster of patient. So like this, we use this method and in our COVID group, and we identify 4 clusters of patient. One cluster 1 is mostly the mild patient, hospitalized mild, and cluster 3 is mostly the severe patient, and cluster 4 is mostly the COVID-19 negative, PCR negative. But cluster 2 is both mixed because the immune response may not be initiated because we collected the samples in the beginning. So we excluded this patient because these clusters of data. And how they move like this. And we also look around 18 interleukins, and then we did a scaled interleukins in this patient, and you can clearly see the cluster 2 is actually, there is some 2 outliers, and cluster 1 is mainly mild moderate, cluster 3 is severe, and cluster 4 is healthy. So going from that, we also developed a personalized genome scale metabolic model. So what is that? So we have a metabolic atlas, and actually this is the developer of this is Professor Adil Mardinglo, who is developed along with other Swedish collaborator, this human metabolic atlas. So here there are several metabolic stacks. So like 57 metabolic stacks are there, and one can see how the reactions are happening. So for example, giving a pictorial one, so we have a metabolite here, gene here, and a metabolite here. So gene 1 is red, so it's up. Gene 2 is green, so this is also up. But gene 3 is low. So these reactions, because metabolic reaction can happen in any direction, is mostly happening in this direction to find out the metabolite here. What we did here also we used the viral biomass function. So when a virus go inside our body, how it utilized the ATP to ADP ATP hydrolysis, mainly that reaction. And then based on that, we first developed a group specific. We only considered on 3 groups where there is no mixing of patient. And then we go for personalized genome scale metabolic model. So after doing this, we try to identify, is there any specific immune signature, those who are severe. So one is mannose, which is our biomarker. So these clusters of genes and metabolite, we term that the severity specific network. And when we look, what are the pathways belongs to these clusters of gene, they are mainly the same: HIF1 signaling, Rig-like receptor signaling, NOD-like, those are antivirals, and also there are some chemokine signaling pathway that are there, complement and coagulation cascade are there. So there's a top path which we already know, and including the insulin signaling pathway. And then we try to identify, is there any very COVID severity specific pathway that is working, or COVID specific pathway. So what we have observed, the flow of metabolite are mainly restricted to this TCA cycle intermediate, so which is modulated by the transporter like SLC25A, SLC25A10, A1, and A11. And these, when we use the single cell transcriptomics data, these are mainly expressed both in COVID patient and healthy control by the monocyte. And then we try to identify gene and metabolite essentiality analysis, and we identified some genes that are only COVID-19 specific, but we are not able to distinguish between severity specific because of the sample size. So one of the features was this alpha ketoglutarate, which is, if you see here, number 2 and number 4, which are mainly the healthy group, nothing, but is high in most of the COVID patients. And then we did a single cell type metabolomics, so where we measure the TCA cycle product and amino acid, and you can see in the monocyte higher level of alpha ketoglutarate, malate, and citrate, so which give that there is a metabolic dysfunction. inside the monocyte. So which we are presently looking, like which subset of monocyte and circulating monocyte are changing, or the different metabolomics profile using a single cell metabolic analysis. In the next study, because system level metabolomic study is also influenced by the gut microbiota, because it has been shown that SARS-CoV-2 depletes CD4 and CD8. So and some patient has diarrhea as a thing. So this study is actually from Switzerland and Cork. So there are 4 hospitals, and we have gone for mild, moderate, we combined together, then severe, who survived, and severe death, unfortunately, who died off. And we did plasma metabolomic study, and it's obvious there is a difference metabolic profile, but the few things that are which are very striking to us is a mannose. So you can see a gradual increase, but also tryptophan pathway. So there's a gradual decrease in tryptophan and increase in kyanulate. And as we are, my group is mainly interested how these different metabolite genes are acting together rather than looking for single genes and metabolite. So we did a weighted coexpression network, and we see that most of the microbiome derived metabolite, that is primary bile acid, secondary bile acid, and short chain fatty acid, as distinct in few communities. And then the microbial processed, so that is leucine valine or branched amino acid and tryptophan metabolism, they are in the center. And 2 are the butyrate and valine, they are also in the high. Then when we zoom in, we can see they are tightly associated with each other, and they are interacting with each other in a good way. So I still put a question mark because it's just association. We are looking into depth, what is the specific mechanism behind it. We also have 16A's microbiome data, what we have observed. There is again the similar trend, what we have observed.
Sorry. So
Yeah.
don't you think with change in mutations in the virus, this will also change, or it will be?
No, I don't think so, because this is what we have observed in HIV. We use several different subtypes and other things. There is not much change in this kind of metabolic profile happens. If the virus is changed completely, like from MERS to SARS, it can happen. But within the small changes, up to 30% changes that we observe in HIV, we don't see anything in the HIV. So I don't think that it will change.
That means these can be very good drug targets also, constant for all the variants. What do you think?
Yeah.
Okay.
Yeah, it could be, but as a virologist, I don't want to overestimate our in vitro studies. But yeah, it could be.
Yeah.
It could be possible. So when we did the microbiome thing, without any group severity in mind, we can clearly see a 2 group of microbiome, and then increase in death is there in the microbiome group 1 and cytokine and again in the group 2 compared to mild moderate patient in group 2. So we are actually looking.
Yeah. Just to extend Sanjay's question, I mean, like, for example, the original strain 2020 alpha versus the Omicron, where Omicron, I believe, replicates faster and to a higher degree. Won't this affect the metabolome, especially your mannose and glutamate to glutamine or glutamine to glutamate conversion?
Yeah, because they have, if our prediction, because again, this is our prediction, if these are changing, if the virus is replicating more, it can take more energy from the cell.
Yeah.
And then it could possible the profile what we are seeing in mild moderate case, most probably the profile will be the similar. If I quickly go back, so for example here you see, so this is the mild moderate, so the orange one, so sorry, the blue one, so it could possible that it show similar pattern like mild moderate, but may not be the biomarker for severity because people are not getting severe. Is that
But death is
answer?
increasing. Death is also there. Even with Omicron, death is there, no? So which probably would be because of the severity,
Yeah.
isn't it?
So I think maybe because the metabolic profile is actually very COVID specific or SARS-CoV-2 specific, and there is some biomarker of severity. So in that way, I think the best way to
So
define.
your hypothesis is that it will not change, which is fine. I just
Yeah,
wanted to know if
it will not
change.
change, because maybe most
Okay.
of the Omicron patient will show the profile like what we see in mild and moderate cases.
I see. Okay.
Okay, so in the final study that again we are now continuing, and this is the reason why I'm telling it will not change, because there is this patient who reached to the hospital ICU and survived. We are following them. She's 6 months and 9 months after recovery. So it's this is mostly called the PAX student or post PAX patient, like post acute sequel of COVID-19. To our surprise, those who reached to the ICU, their metabolic profile did not restore. So these controls are actually the very highly head-to-head match control in the same age group. What is striking is this: serotonin, glutamate, quinolate. These are serotonin is low, glutamate and quinolate is high. What they do, because as we see dysregulated metabolism of tryptophan in mild and moderate cases, so it can shut down the serotonin production, and that can actually accumulate the quinolate, and which has a modulatory effect like mood, anxiety, sleep, cognition, and memory, which we have seen in long COVID patients. So again, I put a question mark because this is just 20 patients. The selective serotonin uptake inhibitor, mainly the antidepressant, may be helpful. Those who are very low, because you see this 3 patient has very, very low serotonin level, and actually 1 patient go down despite they recovered from COVID. So that could be one of the option that one can give. So what we're doing further, we are actually doing, because our basic interest is to find out which specific, so that we can develop a very targeted thing, which specific enzymes or protein are working. So we are doing a SAN scanning targeting the central carbon metabolism, and then we'll see how it affect the replication. If replication is stopped, how that affect the metabolic modulation. And we started, hopefully in couple of weeks time, I have the data that to inhibit the glycolysis. Again, here we use glycolysis inhibition using 2-DG as a positive control. I believe the DRDO already have the data. And then the glutaminolysis, that is our target, and see in K18H-ACE2 transgenic mice, and see whether they are surviving or not. And then we will also do multi-organ immune phenotype from brain to gut, because we are interested to know the gut-brain axis in COVID-19. So with this, I'm coming to the conclusions, because what we have in the first phase, we show that SARS-CoV-2 hijacked this PIK3 HEFON signaling, as well as use the central carbon metabolism. And when we block the central carbon metabolism, there is a inhibition of SARS-CoV-2 replication as well as virus production. We have, when we did personalized genome scale metabolic model, we see the monocarboxylate transporter expressed by monocyte and metabolites such as alpha-ketoglutarate, which is actually also a master regulator of immune response, and butyrate, which can be both microbiome derived and host process, crucial play role in COVID-19 severity. We have observed, when we did integrated analysis of microbiome features with cytokine and metabolites, and we have seen microbiome derived immunoregulatory process, which again we are trying to see it in mouse model. We see how far we go. And then the serum level of protective neurotransmitter serotonin were reduced, and higher level of neurotoxic metabolite like quinolate is high. So this actually need attention for neurocognitive impairment in long COVID patient. And this is my group. I have both experimental and bioinformatics. So Shoham, actually we did together PhD in St. John's. So I was that time doing PhD in Karolinska, but I was placed in St. John's, and Shoham was doing from there. Who joined me, and he actually established the all infection model in the lab, and he has several papers on that. And then I have Shubha, who is mainly experienced in the biochemistry. Alejandra, who is experienced in the CRISPR-Cas9 and signal transduction, and Sarah and Mike, who are cell biologists. She is working also on very specific with Shoham for interference response in SARS-CoV-2. And I have two bioinformatician, Anup, who I think now working with me nearly 10 years, who is mainly interested to go for this network-based analysis, and Flora, who is a new PhD student who is working on machine learning. So our major aim to develop genome scale metabolic atlas for RNA virus, which are a potential of emerging and emerging potential. And I have a master student who are working mainly helping in the lab with different assays. And then this kind of thing is not possible with a great collaboration. I mainly worked with HIV, but Ali, I should say that he bring me 3 years before in Crimea Congo hemorrhagic fever. And with Matthew Selberg, they are developing a DNA vaccine where I am helping them to understand when this DNA vaccine go inside the body, how the metabolic reprogramming happen. And I have a collaboration with Adil. It's a new collaboration because they are mainly experienced in cancer. So I want to see, can we develop a model for infectious disease? And all the microbiome aspect is at APC microbiome. So, and in Sweden, we don't have our own lab, but it's a more like a core facilities. So we can use their facility, just book the machine and go. So this National Genome Infrastructure or Swedish Metabolome Center is one of that kind of example. And my major funding is from Swedish Research Council. I got 4 funding from them last 4 years, and also from NIH. So with this, I think I will stop. Yeah, I'm ready to, and thank you for your attention, and I will take questions. Thank you.
So,
Professor Bamzai,
okay.
I think is looking for some question. Dr. Natarajan, please take over. Professor Natarajan.
Okay, I thought you asked like, okay. So,
No.
Fantastic talk, Ujjwal. Lot of
Thank
work,
you.
lot of collaborations, and I hope it will all continue. So it's a virtual clap. Hope you can hear. Of course, like nice of you to still be like active for questions. I have a couple of them. I've already asked a few, so I will leave the rest to the students. Hopefully, my advanced immunology students were participating in the talk today, because I just finished a T-cell metabolomics in the last class. So this could be a nice addition of how it can be like applied in like infectious diseases. So you did mention about like the GLUT1 receptors in CD8s. Of course, CTLs are important, but for antibody-mediated immunity, CD4s are also very crucial. So were you able to look at GLUT1 levels in CD4s as well?
Actually, not that much, because these antibody are not very specific. We tried 4 different antibodies, okay, and actually none of them. It's shown CD8 very nicely. CD4, yes, but it's not that good. So I'm not fully confident on the antibody itself, because we got the similar questions from the reviewer also, but there's no good antibody that we can measure. So yeah, I'm very much confident on XCT, but not in the GLUT1, that antibody.
How about transcript levels? You can look at
transcript level is quite high, actually.
It's quite high.
But we have not checked in the single cell level because we don't yet done. So the single cell transcriptomics data I have shown, it's from a Chinese group, and we have the similar group like category like healthy, and there we can see the level. But I can actually, it's a very good question. I will look all the GLUT and I can share with you. I'll send it to
Sure.
Sanjay.
Happy to.
So,
Happy to. The other only last question I have is I've already taken too much time. Were you able to score for TGF beta levels also? Because you do
no,
looked at the interleukin panels. TGF beta
no.
and the TH17, those are IL-17. These are the ones that really interest me in COVID.
Yeah, so TGF beta, we are now looking into using the ELISA, but that method that we use is not, it's like a 92 protein, just 1 microliter. So that's we didn't, but yeah, Shoham is actually looking for TGF beta using ELISA, which is more sensitive and we can use more amount of plasma.
Okay. Okay. Very nice. So if there are any other questions, I'm sure Ujjwal would be more than happy to answer. I leave the floor open. Sanjay, maybe you can coordinate this because you've been typing it in the chat.
Yes, sir. So far, I could not see any other questions. Students, please be curious. Thank you, Ujjwal. It was so nice question.
I think
sorry.
Professor Bamzai had some questions.
Yeah.
No, just Ujjwal, fantastic. It was a treat to the ears and virtually to the eyes. So I just wanted to, it's a query, very general one. We have this metabolic reprogramming, which seems to be overlapping in terms of certain networks and pathways between unrelated diseases. So what is your take on that once one has to finally come out with filters to pick up the specific candidates? Like you talked about the pathways, I find between cancer and this COVID-19, SARS-CoV-2 situation, there were many overlaps. So what's your take on that?
Yeah, it's a very good question, because what we have most of the time, we see that just go by the p-value. So that's why in the beginning itself, I say that I do not look much for p-values. Because when we use this database was mainly developed for the cancers. And then we are using these for infectious disease. So it's very like when you do the gene set enrichment analysis or anything pathway, so that is highly overrepresented by the this cancer related pathway or diabetes related pathway. So what we have actually developed, we developed our own database looking into factors in the infectious disease. I think in the first paper we give that, we open the database. So that's mainly the cytokine related response. And then from there, we try to do this network analysis to find out how they are co-expressing together. So we have our own database, and we now data mine actually 5 different databases and make a data set specific for infectious disease. We will hopefully publish soon that database. So there we can filter out. But when in the acute phase of viral infection, there's several things we have also observed in HIV and other. There's lots of similarity in terms of with cancer, because this Warburg effect in cancer, that is the major pathway that virus, specifically RNA virus, also use, that HIF1 mTOR signaling, which is also a central pathway for cancer as well as lifestyle. So my major interest actually is to identify the biological aging mechanism in chronic disease, as well as in the COVID-19, those who went to ICU. So we started this cohort. We have a plan to follow them up for 5 years to see these transient metabolic changes, what happened during the acute phase, how much time it takes to normalize. And then rather than drug, maybe some food itself can improve this. So that's my way of thinking, because we can use antioxidant food. Simple food can improve, because for example, serotonin we can get it from several food. So that way it can like transient change in the acute phase of infection can be related with cancer, but it should like trade off. It should go off after certain time when the people are normal stage or healthy. So hopefully we can see it in COVID-19, this our long COVID cohort. But we need to know how much time it is taking to come down to a normal level.
at any point in during your study, which is huge, did you find LKB AMPK pathway involvement?
AMPK is involved in COVID-19
Uh,
because this is the start of the HIF1 signaling. So
okay. So
it's
I was
there.
no, but I was deviating it a little bit towards the glycolytic pathway and focusing on my interest on pyruvate kinase. So PKM2, PKM1 shifts. Anyway, I just wanted another query which I had. During the filtering out process of the candidates, if suppose are you losing out on some candidates where weightage is not that great, and then obviously because the program is written that way, and you lose out on them, but they have a combined effect. Maybe they have a synergistic effect. So what do you say about that?
Yeah, so generally we go, there's a high chance of losing out the small interaction. But as we go for weighted co-expression and the community, the central community, the link with the other community will not be that great. So most of the genes, even it's lowly expressed, will come in the central community. We have seen that one. But generally what we do, so that is the first stage, that we do the higher weightage. Then we took those pathway and choose the other genes and see, just filtering out only those many genes where we may have say adjusted p-value a little bit lower. So we go low, we go different stages, not to lose out any genes. But also these genes, part of the pathway, are variable in different databases. So that also we try to pull first and then use our own terminology like that.
Incidentally, we have seen that when insulin-driven pathway is there, PI3 kinase, mTOR, HIF1 alpha would be activated, but ROS would be that will play a very contrary role. And mitochondria would obviously be involved over there. So any idea about that? I won't take this question further and leave it here. Thanking you, Ujjwal. This was wonderful. And leave it for students to ask questions, and maybe Damanji.
Okay.
Thank you, Dr. Ujjwal. It was a wonderful talk and great learning, especially for me who is not working in system biology per se, but a little bit on cancer biology. Yes, you are very right. If pathway is getting modulated in almost all the diseases, infectious or non-infectious. So when you were talking about your severe cases or mild and moderate cases, these patients must be under treatment, right? And
No,
the treatment
so
could be, they were not,
no,
okay.
no.
You
So
took
they
it on
are
day
collected
one.
day one. Yeah, day 1 to 2. Before they got any treatment, we collected. So that's why it took us long time to recruit only 12 patients.
Okay,
So they are
because
collected in day 1.
normally people, once they start getting fever, people start giving them paracetamol and steroids and other things. So
No, we do not actually do here in Sweden. So you cannot buy
okay.
those drugs. Paracetamol, yes, paracetamol will be there, but we have not seen any paracetamol traces in
Okay.
the metabolomics data. But generally, like here, the trend is like, even for me or my son, we wait for the fever to go down by themselves. When it crosses like 104, then only we give paracetamol. So, and here you
I
will not
see.
get any steroid and any drugs outside. For us, for fever, cough, cold, even cough medicines are not taken. It's only paracetamol. That also, like, recommendation is like, if it goes past
So
like
all
103.
your cases were, although you collected from different cohorts were from different places.
Yeah,
So all
we
of them were
tried.
like that.
Yeah.
Okay, all right. So that's really good, because otherwise you will see a lot of effect of, because in India I remember on day 3 they start giving steroids and then
No. Here only if you reach to ICU, they will give something. Until then,
I
they
see.
do not give anything.
I see. Okay,
So
great.
even they try to do, like, for initial patient, they just giving like oxygen, and
Okay.
to some extent that also like amino acid and glucose when they're not able to eat.
I
So
see.
our mild cases, though they are in hospital, they are all eating, so there's no drip on them.
Okay. All right. Thank you.
So two more faculties have questions. Aparna ma'am, please go ahead. Directly you can ask questions.
Hello, Dr. Ujjwal. This is Dr. Aparna from ACBR. I'm a faculty here. I had just two questions. One of them is like, since we are trying to inhibit probably aerobic glycolysis to cope up with the COVID. So, what is your opinion about keto diet in this case? Is it going to be having some effect?
This is actually a lot debated in Sweden. I think those who are on keto diet, they are doing better compared to those who have other thing. But yeah, I don't want to, because COVID is such a way, I don't want to like predict something based on our data, because this is just a one cohort data. But yeah, certainly diet health has good effect. As I see here in Sweden, we never had any lockdown. Generally, the people here are very healthy. They are very health conscious, and they go for jogging. And what we have seen, there was a recommendation in the beginning, because there's no lockdown, that don't go to the shopping complex or indoor games. And then there's a 400% increase in individuals or the people here in Sweden to go to like in the park or in the open field. So that certainly good health is always for viral diseases is always, you can take care of it
Right.
quite well. So
because ultimately your group is trying to target the metabolic pathways, the metabolism that can suppress COVID infection probably. Yeah. The other question is, because you were talking about the tryptophan metabolism, altered tryptophan metabolism. In one case, you found serotonin is high, whereas the quinolinate, which is a neurotoxic thing, neurotransmitter, it is high. Have you looked at the other tryptophan metabolite, which is kynurenic acid, which is neuroprotective in nature? Because you talked about serotonin, you talked about quinolinate, but kynurenic acid is something, because it's basically the balance between kynurenate and quinolinic acid, which is, if altered, will lead to either neurotoxicity or neuroprotective thing. So since it wasn't mentioned, so if anybody has looked at it.
Yeah, we looked into this paper only. I just show one slide, so maybe I can send you. So yeah, it's altered severely.
So
this KD ratio.
Okay, the balance between kyna and quinolinic acid. Okay, because microbiota do affect this balance. Right. Okay, thank you so much.
Okay, Madhu ma'am is also having question, I guess. Ma'am, please.
Thank you, Sanjay. Ujjwal, it was a very nice talk, very elusive and concise. I have one question that we know that increased metabolite, not metabolite per se, but the increased these glutamine requirement is there for immune cell function also. So, and this also occurs along with the high glutamine utilization by host cells in virus infected individuals. So how do we develop antiviral drugs? Specifically, we just want to target the glutamine metabolism, and how it can be developed as a therapy then, because otherwise there is a requirement also for patients or cleaning out the immune functions properly. So how do we target this pathway?
Yeah, so what this target, like glutaminolysis or glycolysis, it's the host-directed therapy. Because see, for any viral infection, particularly this kind of virus, 85% of patients don't need any treatment. If they have good immunity, they will pass through it. So this treatment, the way the 2DG is used, is only for severe patient, and it's a very short. If you can stop the viral reproduction, viral replication, and then try to do with other. So this could be a supporting for the severe patient who are inside the ICU or somewhere there, not for everyone. So it should not be considered for everyone. The way 2DG also used is only for the severe patient. And it's mainly host-directed therapy. Short-term therapy, just stop the virus replication, and then you can take care of our immune system can take care of the virus or the recovery. And second thing, when these drugs are being used, they do not do exact complete shutdown. So these are partial. So you should have a dose which do not do, then the cells will die inside the host cell. So you have to have a dose that is enough to take care of the immune system or the cells to grow, but not in that way that all people start taking. It should be only restricted to the severe patient who don't have any other option to save their life.
Okay.
Okay. Sounds good enough, but the problem in India, everyone will start searching for this in initial stages itself. That is the problem here, and very severe. Last time in last wave, I know several of the steroids were also over prescribed even by doctors. It's not that they were sold over the counter. Doctors even prescribed on third day itself, start taking even though there was no symptoms of oxygen depletion or oxygen requirement. Then also doctors saying start taking precautionary steroids. So there was a very overuse of drugs in India here.
So that could be.
It's very cautiously. Even when 2DG was announced, the people were saying let's start buying it and keeping it in our almirahs, and then we will be having when we have some. So it's self-medication is a very, very severe problem in India. So that's why.
Yeah, and I will tell my experience in Sweden after last 6-7 years. For my own personal experience, for my son, apart from paracetamol, I never give him anything for last 6 years. And it's very important that these children, particular children, they are also prescribing steroid, which is very, very bad. And we do not give antibiotic. Getting antibiotic here, doctor has to write why they are giving. There is the other option too. So that's why I said, like, here getting a drugs is very difficult in the over the counter, and also like when you go to ICU, there maybe you will get some drug, but still it's they try to heal it naturally. I think that thing need to be changed in India, like just prescribing. Even like you don't need paracetamol in first 3 days if you are not like 104 and 5 fever. So here I give my son paracetamol when it passed 40. So that is 103. Until then, just take care. The normal body, our body will take care of it. And also high temperature is good. The virus can be killed because of the high temperature.
True. Yeah. Thank you, Ujjwal. So our another faculty, Professor Pratibha ma'am, you can ask your question directly, please.
Thank you so much, Dr. Sanjay. I'm not able to open my video. Otherwise, I would have opened I don't know, I've been trying, but it is somehow not working. But anyway, it was very nice talk. I was all throughout this talk in there, and it was nice. I'm going to have, because Dr. Madhu has asked the questions which I thought I'll ask, but I have still one question, probably. Since, we all know now this virus is getting all the time mutated. We know that, it's been doing it again and again. So do you think that a development of a drug for this particular kind of virus, which has been changing again and again, will be worthwhile?
Yeah, so that's a very good question, and I agree with you, because if you remember Tamiflu, there is another drug which has been used heavily.
Yeah,
So I think it's very difficult to go, and that's why my research, I drive it towards the host directed, so that it target the host. And we just may need this kind of treatment for 2-3 days if it is host directed, because if we build that kind of immunity with maybe small supporting treatment in the beginning, then our body can take care of it. So yeah, it may be short term is good, but I'm not sure this kind of virus which mutate that we can do that good antivirals.
Yeah, even if we are talking about the host directed, if we go for the Ayurveda, it says every person is different. So again, it is not that easy.
Yeah, so you can see this kind of virus, 85% of.
Yeah,
Yeah,
HIV,
85% of
yes.
the individual will naturally heal. You don't need any treatment for
True.
virus.
Yes,
So, and
true.
those who reach to the ICU may need some treatment or in the hospital. So I don't think this virus, like over the counter, this drug should not be given, like particularly this kind of virus. Then it will be more complex because then the virus will change by themselves. And then afterwards, you don't know what kind of mutation will give the rise to them. So this, whatever the antivirals or this host target therapy is only for say 3% of the patient who are 15%, 3% of those 15% who reach to the hospital. And most of the time our body will take care of it.
Sure. Yeah. So the best thing is to keep and maintain your immunity.
Yes. And good health.
Yes,
Good health, good food.
True. So thank you so much, Dr. Ujjwal. Very nice talk. And it's a really nice because people get excited today. I mean, my real brother got COVID infection, and the family is in just like running here to there. Then I said them, okay, if he's all right, stay back home, and that's it.
Just
So,
water and orange juice. Water
Yes.
and orange juice is enough.
Yes.
Yeah, that's great. Yeah.
Because you need to have vitamin C, so
Yeah.
orange juice and water, warm water and this thing. Yep, sure.
Yes, ma'am. You too take care. Yeah,
Thank you
while
so
you are
much.
traveling. Yes.
Thank
So,
you, Dr.
Dr.
Sanjay.
Ujjwal,
Very nice, good organized seminar. So nice
you,
of
ma'am.
you. Yeah.
Thank you,
And
ma'am.
keep on poking to all of us. Okay, be there. So nice of you, Dr. Sanjay.
Sure.
Thank you so much.
Dr. Ujjwal, if you don't mind, can we ask few more questions from the students?
Yeah, go ahead. No problem.
Yeah. So one of our students, Suas, is asking, why do you think gamma delta T cells were down in patients, while otherwise they regulate viral infections responses amongst other features?
Yeah, this is because their immune system is exhausted. So there's nothing left to respond. That's why they might have reached to the ICU, because they are not able to, the function of gamma delta T cells to counter the virus is gone. So that's why they might be exhausted and trade off from the body.
Thank you, Ujjwal. Suhas, I hope you got your answer. So another student, Kapila Kumar, is asking, in long COVID complications, are we taking into account drug environment parameters, as metabolic changes are very dynamic in nature? Whenever you are taking a drug, probably the metabolic environment will also change.
Yeah, for long COVID, as of now, apart from giving antidepressant, I don't think any treatment is recommended for long COVID. So, and in Sweden, it's mainly the counseling that's happening, because it's very difficult to get antidepressant drug for. So I don't think in our patient, we are considering those and we are putting those drug in the model, but not that way, because there's no specific treatment for long COVID as of now.
And Ujjwal,
Apart from boosting your immunity.
What should be the duration for long COVID if you want to consider?
As of now, like as we show in now, so we have like 9-month-old long COVID patient. So
okay.
So it could be more.
Thank you. Another question from Shivani Panchal. She is asking, among amino acid, why only glutamate metabolism is getting affected? I think it is already answered. But anyway.
Yeah, no, it's not all amino acid metabolism is affected. Glutamate metabolism is high. So glutaminolysis is high. Rest of the amino acids has gone down.
And any other amino acid can also be affected? She is asking.
Yeah, all the amino acid actually gone down, because virus is using those amino acid only to get its own genome.
Wow.
So most of the amino acid are low, except the glutamate.
Okay.
Even glutamine is also low. So that's why it's called, the process is glutaminolysis, because it's more utilization of glutamine.
Okay. Thanks, Ujjwal. One last question from another student, Sarika. She's saying, very nice talk, sir. I have one question. Why you have focused on GLUT1 transporter? Because many other transporter like GLUT4 is also related to SLC2A family.
Yeah, so GLUT1 is actually ubiquitous, and you can find it in most of the cells. That is one of the reason, because all GLUT is not expressed in blood cells. That is one thing. Second thing is the antibody, though people are working getting good antibody for FACS is difficult. There are antibody for Western blot, but that will not tell you it is a surface or not. So that's the reason, because GLUT can also internalize and go inside.
Thank you, Ujjwal, and thank you everyone for asking very nice questions. So, Director ma'am, if you give us permission, then we can go for vote of thanks.
Sure, Sanjay, please go ahead.
Okay. So we are at the end of the meeting. So it was very nice journey, and you have given beautiful talk, Dr. Ujjwal. I must also thank almost all 70 participants who are present during the entire talk, and they are from different institutes in India and also from abroad. And first of all, I'll start the vote of thanks by thanking our speaker, Dr. Ujjwal Neogi, who is from the famous institute, KAROLINSKA Institute from Stockholm, Sweden. And why I am so excited about him, because this is the institute who usually decides who will be given the Nobel Prize in physiology or medicine every year. So I am really excited to also start our collaborative work with you. And I must say that our other faculties will also be interested to collaborate with you because of your expertise and having immense amount of facilities with your lab. Okay. Next, I will also like to thank our chief guest, Professor R.N.K. Bamjai, for taking out of your very busy schedule and blessing us with your expert comments, etc. And we hope to see you again with us. Thank you so much, sir. I would also like to thank our chairperson, Professor K. Natarajan, sir, for giving his insightful comments and asking so many stimulating questions, because it was so relevant to have you as the chairperson. You are the expert in the field and asked so many good questions and made the platform very interactive. Thank you so much, sir. And I'm also thankful to our director, ma'am, for helping us to organize this seminar series. And it is, I think, going well for the students, especially. They are also coming to know what kind of research is taking place across the world and also in India. Also, I'd like to thank all the faculty members from our ACBR who has also asked so important questions and vibrantly present during the entire meeting. It also helped and inspired our students for sure that we have been organizing this thing even during this pandemic while people are so much stressed. Also, I'd like to thank all other students, postdocs, research scholars for their active participation and asking questions. I'd also like to thank my students from my lab and also Master Rajat, who is Dr. Meenakshi's son, who is taking care of the electronic items, including YouTube streaming. Thank you so much, Rajat, for doing it once again. With this, I'd like to tell you that we'll be having similar kind of talk every month, and hopefully you will all join it and enjoy it. With this, I'll also wish you a happy and safe Republic Day in advance. Have a nice day. Thank you, everyone.
Thank you.
Thank you, Sanjay. Thank you very much.
Thank you, Sanjay. Thank you for organizing.
Thank you, Sanjay.
Thank you.
Thank you, Dr. Ujjwal, Professor Bamjai, Professor Natarajan.
Thank you.
Thank you, everybody.
Thank you. Thank you, everyone. Thank you.
Thank you, sir. Thank you, sir.